Well, good morning, everyone, and welcome to our EMX 2025. I'd like to also greet several new friends I made at the Cryo-EM meeting at the weekend, co-organized by Wa and his people, which was terrific. Now, normally at this time of year, it's a time of hope and aspiration. But this year, forgive me if I remark on the continuing conflicts in Eastern Europe and uncertainties in the Middle East, the stark manifestation of climate change, which we see a few hundred miles south of here in California, which is causing incredible devastation, and of course, possible future travel restrictions and who knows what from the upcoming US administration. It struck me then that this a forum like this, whereby we join physical and biological electron microscopy community, we scientists speak the truth. This is more important this year than perhaps any other time since the pandemic days, early COVID days, when we started this meeting. So we will continue it in that spirit to bring us together as a community and to share our thoughts across nations and look for a better future over the course of this and the future years. Having said this, we have another exciting program today. The physical sciences speaker who will go second will be introduced by our incoming faculty member, Kate Reedy, and I'll introduce her at the time. And right now, I'm happy to hand over the forum to our friend Hua Chu, who will say a few remarks and introduce our biological speaker, Michael Elbaum from the Weizmann Institute in Israel. Hua, please.
Hello, good morning, everyone, and Happy New Year's. And I echo what Bob has said. I think the original idea of this symposium is to unify the investigator who are interested in electron microscopy across the nation. So I think we hold that spirit, and I think regardless what, I think we will stick together and continue to push the frontier of electron microscopy in its own development as well in its biological and material science applications. Today, I'm delighted to have Michael Elbaum from Weizmann Institute of Science and in Israel. He is currently the head of the Department of Chemical and Biological Physics. I have the fortune of meeting Michael last year in person, and he is a very personal individual and extremely knowledgeable in a lot of techniques in electron imaging. And he has been active in development of a number of imaging techniques and also its scientific applications. And in particular, in the area of soft X-ray, cryotomography, and cryoscanning transmission electron tomography. And in the recent years, he has put his effort in extending the cryo-EM to a cellular scale. And his lab combines approaches from materials and life sciences, and recently has introduced the automated cryotomography using the 4D stem. And it's my great pleasure of having Michael in the late afternoon in Israel to talk to us about this exciting science that he and his colleague has been doing at the Weizmann. Michael, welcome.
Thank you. Thank you. Do you hear me well?
Well,
you hear? Very
yes,
good.
very well.
So I'm really delighted and honored to be in this forum. I have to say it's my favorite web seminar, and the spirit of combining the materials and life science together. I think that's something that we've had for some time, for a long time at Weizmann, and it's something I really applaud, that people have lunch together and they can discuss ideas. So this is a project that began in a collaboration with Sharon Wolf and Lothar Huben, again, life science, material science, and the recent developments involve very heavily Shachar Seifer who works in my lab. So I'm going to start with the teaser. This is actually the last slide, and you'll see where we're going. So these are T4 bacteriophages, and the life scientists will recognize all of these features. We'll see them in more detail later. But this is I want to emphasize this is a cryotomogram. So this is here we see all these fine features, and this is where we're heading. But we're going to start with somewhere else. Most of the talk is focused on technique, and nonetheless, I want to start with one proper biological motivation. And this is a recent work we've done with Leslie Lazarovich, who really inspired this for quite a few years. The topic is malaria, the parasite that causes malaria. And as we know, the parasite, Plasmodium, digests the hemoglobin in our red blood cells. As it digests the hemoglobin, it releases the heme, and the heme itself is toxic. It forms crystals. And these crystals are called hemazoin. They were long called the malaria pigment, and they were the way that the field doctors could recognize that you have malaria. The anti-malarial drugs we take actually inhibit this crystallization. The crystallization takes place in a vacuole called a digestive vacuole, and here we can see a cryotomogram of the digestive vacuole. And we see the crystals inside a bag, a membrane bag. They seem to be nucleated on the surface, and there are large ones collecting in the center. We also notice that they're blinking. So why are they blinking? Because they are diffracting. And this is a diffraction contrast that we see very nicely in STEM when the crystal matches the zone axis. The crystal orientation matches the zone axis. We can also see the crystals in reconstruction. So here we see them reconstructed in 3D from the tomogram, and you see the crystals very nicely, but they have this bizarre shape, this very curious shape. You see a kind of a handle. Do you see my mouse? Do you see my, yes. So you see this handle. They look like a kitchen cleaver. So why are the two ends asymmetric? This was a kind of a puzzle that motivated a study by electron diffraction. So now we move to the right side, to the diffraction mode. So here's a crystal, an isolated crystal on a grid, and this crystal is tilting, and we see the reflections from the tilt, and here we see the Bragg reflections. So we see the Bragg spots, and we also see the diffuse scattering, diffuse coherent reflection from the water, from the vitrified ice. When we put it all together, we could understand the bizarre shape of the crystals as a reflection of the isomeric content. These heme moieties form dimers. The dimers can form in different ways, and the crystal is populated by a particular subset of the possible dimers. So we could see the diffraction gave us information on the isomeric content, and the morphology gave us information on the shape and the orientation of the crystals. Putting these together, we could move between a scale close to microns to a scale close to angstroms. So this is a kind of motivation for the two methods. And I want to show, first of all, the similarity between the methods. So what do they have in common? Both of them are diffraction techniques. The microscope works in diffraction mode. Both reconstruct a Cartesian model from a tilt series, and both are unconventional approaches in cryo-EM, or applications in cryo-EM. How do they differ? The STEM is scanning a focused probe in 2D and then integrating over the scanning. The electron diffraction is looking only at one point in real space. It's covering a patch in real space, and we're looking in 2D at the diffraction space. So this is crystallography. So STEM, we can say, is 2D in real space, 0D, 1 point in diffraction space. Electron diffraction is 2D in diffraction space, but only 1 point in real space. Of course, we can combine these and make a kind of a nano diffraction, and this is leading us to 4D STEM, 2D in real space, 2D in diffraction space, and we want to add tomography to add one more dimension. This can be the tilt dimension, and the tilt dimension we convert to the Cartesian, to the depth. Okay, back to Optics 101, diffraction or direct or diffraction mode. So what does an objective do? This that doesn't really have to do with electron microscopy. The task of an objective is to map with magnification a point on the specimen to a point in the image. So the blue point matches to the blue point, the red to the red, the green to the green, and we have a magnified image. Since we're looking at transparent objects, we have to play some tricks. We have to play by, we have to work with phase contrast or with interference, which we gain by defocus, or we use a faceplate in order to gain focus to gain contrast in focus. And the task of the TEM, this is really the art of the cryo-EM, is to look at transparent objects, of course. In the direct mode, the projection system magnifies the primary image. But let's look here. We have this important plane behind, the back focal plane, and this is where the rays of the same angle pass through the same point. So no matter where they came from in the specimen, rays leaving at a certain angle pass through this point. This is the plane where we see the Bragg scattering. And in the STEM and the electron diffraction, the projection system magnifies the back focal plane, not the image plane. And we have here the spirit of Mr. Fourier living strong with us. Okay, so I call this sometimes scanning diffraction electron microscopy. And what we're doing is to integrate over the diffraction plane. So here we have as an introduction, here is, we pull out the sample, we have the vacuum. We put in the sample, we scatter intensities. So we can collect for the high angles, we call this high angle dark field. Because we have a bright spot on a dark background. We can look at the bright field and see what's missing from the original direction. And we can also play with the selection of angles to have a smaller angle set or an annulus within the bright field. We can look at a crystal, and then this is what we had before. We put a detector here at the zero order beam, and we get diffraction contrast. And we can look importantly at phase gradients. So here we see when the beam passes through some disturbance in the projected potential, some difference in the matter, then the beam is slightly deflected, or the intensities, to be more precise, the intensities are redistributed, and we can pick this up on a position sensitive detector, for example, on a quadrant diode. And the difference, the imbalance of the signal in the detector gives us the gradient in the phase. If we put a grid here, then we can measure somewhat more accurately. We can measure the center of mass, as it's called, the first moment of the distribution, and again extract the phase gradients. And we can go all the way to tachography, looking at the bright field interference pattern, to reconstruct the illuminated structures. So in any case, we need to generate a signal for the representation. In the end, we're going to look at an image. We have to see what we're looking at. Okay, now we should ask, for life science, main question, why bother? So cryo-EM we know works very well. Single particle is astounding. The single particle methods are astounding. These are extending to tomography. What does STEM get us? First of all, we can apply different methods, spectroscopies, diffraction, as I showed. We can aim the beam at an area and look at nanodiffraction. We can look at scattering analysis. And what I want to focus on today is combining the capabilities for phase imaging by 4D STEM. What else is it giving us? When we look at thick samples, we have a problem with the conventional mode of cryo-EM, which is that we try to form a wide field image, but we have energy losses or inelastic scattering as the beam passes through the specimen. So you can see here the illumination energy, the initial energy, and we see loss processes that divert the transmitted signal to lower energies. Lower energies means lower velocity. Lower velocity means we change the focal length of the objective, and then we're not going to focus in the same plane. So the limitation, primary limitation of the conventional mode of wide field imaging is this inelastic scattering, which becomes severe as the sample becomes thicker. So we normally apply an energy filter, and then everything works again. But of course, we lost most of the signal if we apply a narrow band in the energy filter. So we can kind of skirt around this problem by avoiding the formation of a real image in the first place, and this we do with STEM. This has an immediate advantage for thick specimens, for tomography, for ultrastructure, and in particular for study of unique features. So if we want to look at a ribosome and we can average many copies, this is fine. If we want to look at a mitochondrion, it's very hard to average a mitochondrion. So the idea is to extend the capabilities of cryo-EM toward ultrastructure using the relative insensitivity of the STEM method to this chromatic aberration, to this energy loss process. And then once we have a signal, we can apply the standard methods of tomography to reconstruct. Okay. The sad news is that we never have enough data, so we have to apply some more tricks. We can use prior information to remove much of the noise in the tomogram, and now we can see the improvements in a thick specimen. The comfort zone is about 600 nanometers. We go well easily up to a micron, and here you see left and right the improvement in the denoising, particularly for the perpendicular planes, particularly for these planes as opposed to these. So most of the noise in these planes is actually projected contrast from distant planes, from distant horizontal planes. Okay, now one of the exciting discoveries here was these large-scale structures. This is with John Sidat at UCSF. Large-scale structures, kind of slinky structures in chromatin, and this is something we try and plan to pursue further. Okay. One last topic on the conventional cryostem is we can extend to dual axis. This is Peter's work. Dual axis gives us, after the deconvolution, practically an isotropic resolution in the 3 dimensions. You can see here this feature, this microtubule, disappears if we look only at one axis. Okay, so now we come to the new development. So this is our sandbox microscope. Since the fancy machines arrived, practically nobody wants to work on it. So we have more time, and we can be a bit aggressive with the hardware. And Shachar he replaced or rebuilt the scan generating system, the scan waveform system, and we put on detectors underneath. So here is a diode detector from a local company, Elbaum. And this is giving us these 6 segments that we can from which we can make an image. Okay, so one of the first things we did was to put in not a cryo sample, but a set of nanotubes, and this was quite fortuitous. So we can look at them in dark field or in bright field by changing the projector magnification, and we can see them in projection. Here, 1, 2, 3, 4. I'm just going around the 4 quadrant segments, and you see these bright spots are Bragg reflections from lattice strains. These are you see nothing too exciting is happening here, but when we do this with the bright field, you see them belly dancing. You see this kind of belly dancing, and actually these on top are belly dancing a lot. And if you look at this one underneath, it's hardly moving at all. So this is a head scratcher. And actually what we're looking at is the parallax effect. We're looking from different angles. And so we normally think we look with our 2 eyes, basically, back up at the same source. And here we can see another example. And if they're shifting a little bit, the obvious thing to do on the computer is to deshift them. And we see when we deshift them, we can get an extended depth of field. So it's like we've corrected or accommodated the aberration, which we call defocus. We've corrected the defocus in a way. And this is actually a simplification of methods that have been described subsequently for this tilt-corrected bright field imaging using the 4D STEM, the parallax correction also there. So the next thing to do is to consider the differential phase contrast. So as I said, the difference between half spaces gives us a gradient of the phase. And if we integrate the gradient, we get the integrated DPC, IDPC. The go-to reference is here. And of course, we'll try and deshift it. So we deshift the DPC, and then we have a lot of fun. I don't want to go into theory, but we can say we go beyond the weak phase object approximation, which is really a major, I think, a major hurdle in the cryo-EM concept of how do we consider thick objects which don't really match to the limits of the weak phase object approximation. So if we correct the parallax, then we get something which looks like the phase. If we take the remainder, we'll see we get something that looks like the depth. And we can also differentiate in this image shift, which is like taking two images with different focus. So here we see extended depth of field for the DPC by deshifting. And we see here this depth contrast. You see immediately who's on top by the remainder, the DPC2. Okay, we want to do something with an interesting biological sample. This is a recent work, not published, but I think perhaps the most exciting. This is a collaboration with the lab of Florian Schur. They sent us fib lift-out lamellae. So this is the very edge of the state of the art in sample preparation. And what they have here is kind of a layer cake of cells with extracellular matrix, cells with layers of collagen fibers, cell ECM, cell ECM, cell ECM. And this is from their paper. So we put this Peter actually into the microscope, and here we can see cell ECM and cell. The thickness here is about 450 nanometers, almost half a micron. And if we play the movie, we can see here these long fibers, and you see there are two sets of fibers, one running more or less parallel to the surface and one running at a steep angle to the surface. We see also the nuclear membrane here, and we can see also these twisties, slinkies inside. Okay, now this sandbox microscope is challenging to operate. We have a DPC detector, a quadrant detector from Thermo on the Arctica microscope. This is called the Panther detector. It doesn't work quite out of the box, but it's something that serial EM can handle if we work with a bit of scripting. Again, we see a layer cake: cell ECM, cell ECM, cell ECM, cell. And then we're joined by Sharon and Netta Versano, who did this beautiful segmentation. Here we go. You can see these filaments. The MPEG is having some trouble, I think. So let's look at it frame by frame. Here you see this interpretation where we have the parallel filaments and the crossing filaments. And this sample is about 750 nanometers thick. You can see for interpretation the great advantage over a cell of 150, over a sample 150 or 200 nanometers thick. Now we'll move to we'll shift gears. We'll move to a different sample. This is the bacteriophage, and we're using this as kind of a mascot for development. It's a high bar sample. It's still amenable to cryo-EM. We can still do the cryo-TEM, energy filtered cryo-TEM. This is an example from the Cryos microscope with the energy filter. And the question we want to ask is, what can we interpret without averaging? So we're kind of on a borderline sample here. I have to say, it's just a very beautiful sample. It's so encouraging to look at the data and say, wow, look what we see. It's also been very well characterized in the stalk region. The DNA structure inside, if there is any, is not well characterized. So, so we're going to work with this as a sample, and we're going to look now with the 4D STEM camera. This is the ARENA camera pixelated detector from Dectris. There are a number of fast cameras now available. This gives us a rather small pixel array, but with extremely fast readout time. So the time to take a 1K by 1K image is order of 10 seconds. That's not going to challenge a K3 camera or a Falcon 4, but it's a game changer in terms of collecting the diffraction pattern. Okay, so we go through. Over vacuum, we have just a spot in the middle. Over specimen, we get some scattering. We can divide this scattering into rings. What are we going to do with 96 squared individual images? One thing we can do is, is just to integrate over rings. And now at every pixel in real space, we have a vector of 16 elements. And this is not really a new idea, but it's a new implementation. So here we have over the bright field, indicated here. Carlina and Shai are involved in this project at different times. We have the dark field, annular dark field here with the opposite contrast. And now we can look in the bright field. So we can look at the center ring 1, ring 2, ring 3. And I think the material scientists will recognize this as the reciprocity bright field, the reciprocity TEM image, the incoherent bright field, and the annular bright field here. And we can compare this with the ICOM, with what we're supposed to do with the 4D STEM. I want to emphasize here that these are central sections. These are single sections through a tomogram. These are not projections. And I think that the ABF here is perhaps a better representation of a single slice, although the COM has a very attractive texture. Now, we have a vector of 16, so we run it through the PCA code. We have the first component looks like the incoherent. The second component looks like the annular bright field. The third actually has the highest resolution information, the very finest features, and the fourth is the dark field just inverted, and that's verified by the weighting factors that we see in the decomposition. Instead, we can do the parallax differentiation. So here we see this de-shifted sum, and here we see a little bit cleaner, this favorite of ours, this delta IDPC, the parallax differentiated integrated differential phase contrast. So we have now a 4D STEM implementation of phase-sensitive imaging for tomography. And again, this is not a new idea. These come from 1974, again 1978. But with the new hardware, it's now let's say conceptually straightforward, at least, to implement this. So we come to the moment of truth. We've learned to use the quadrant detector for coherent contrast. We learned how to do things from the fancy camera. We really, I think it would be very difficult to optimize conditions without the fancy camera, but do we really need it afterwards? And here we're inspired by works of the Muller and Kourkoutis lab, the late Lena Kourkoutis, so late in NIST. And they demonstrated an upsampling. For us, this was really an eye-opener, an upsampling of the sampled image, of the image. So we can acquire one number of data points, and by working a little bit out of focus, we can upsample into a larger space, a higher resolution space. We do this a little bit differently, but this was, a proof that something is working is a great motivation. So I want to show here. This is Shachar's work. Here we have an image, 512 by 512, of some gold nanoparticles, just plain gold nanoparticles, and we've taken a focus series. So this image is out of focus, no problem. We can put it back into focus by this image shifting. You see it here. but we're still left with 512 by 512. And we have a new method. We call it the shadow montage. And here we've upscaled by a factor of 5. So here we've refocused it, but also enhanced the contrast, and enhanced the resolution. And we're not really cheating. The key is here is to look at a defocus series. This is a different series. So again, a sample of gold on carbon. Oops. And we're going to look at the Ronchigram as we go through focus. Again, material scientists will be familiar with this. Here we are in focus. Here, and as we go away from focus, we have a shadow image. Now, if we do a bit of arithmetic, we can say we have a shadow image at one point, and we'll move over a little bit with the beam and take the shadow image at the next point, and move over and take the shadow images in sequence. And that's the key to this this upsampling. So everything works with beads. I think all the light microscopists know that everything works with fluorescent beads, but you have to try on real data. It's like the deconvolution story. Here's the DIDPC. It's the same image I showed before, but inverted in contrast, just inverted in contrast. And here is this shadow montage, this upsampled image. And now if we have a factor of 4, that's a function of the sampling and the defocus. And here we've gone down to in practice 4 angstrom sampling. So we can focus in here. And here's the movie I showed at the very beginning. And now we can see the T4 phage and look at its structure in detail. We see the capsid with the DNA. Here's an empty one that's already shot out its DNA. We see the base plate. We see the injector. And what I think is most impressive are the fine filaments that you'll see around the stalk. Here you see them wrapping. And here they are. Here they are in parallel. So all these fine features, which we don't normally expect to see in a single image. This is a single data set. So that is, I'll just let it run. That we can also see that much of this noise in the background appears to be filaments, probably broken tail filaments that are left in the sample. So that, I think, is it. This is my final slide, cryostem in 2D, 3D, 4D, and more. I hope I've shown you the power of the tomography. One used to say that STEM doesn't work well with cryo. That's no longer the case. There's a small but growing community, I hope. The primary advantage to my taste for STEM methods is for thick specimens. Thick specimens mean tomography. So we've been focusing on the tomography. I've shown methods for parallax defocus compensation for DPC, the upsampling by the 4D STEM, and the main message is waste no electron. The key for working with cryo specimens for beam sensitive specimens is to use every electron. And with that, I'm done. Thank you.
Wow, that is impressive. Thank you very much, Michael. I think today you convinced me this is a very attractive technologies. Let me start asking, well, there are some questions on the Q&A. Let me ask the first questions. Do you think this will be useful to look at sample with heterogeneous compositions and conformations? Because when I look at your phage, some are empty and some are full, but have you tried to look at something in between?
We're trying to combine this actually with a collection of the dark field scattering. The dark field scattering reveals heavy elements. So we can see small distributions of heavier elements. We worked originally this on calcium phosphate. Calcium is a heavy element for us.
Right.
I'm working now on silica. in silicon in an organic matrix. So in this sense, yes, I'm hoping to have both, the best of both in a sense. This is an extreme case. We see also partially filled phage heads in a different example. I'm happy to discuss that further. I think one of the attractions for me is to be able to analyze the scattering as well as the phase issues, the phase component, and to be able to say something about composition. I would also say lipid we see right away. So lipid droplets, for example, scatter weakly to high angles, more weakly even than the water. So we see a contrast inversion at lipid droplets.
Wow. Okay. So there is a bunch of questions from the audience. Let me start with Martin Halberg. And his question is, what's the algorithm difference between 1C bright field, the TC bright field, and also the shadow montage?
What I missed the first, which bright field? The TC bright field?
The TC bright field.
So the TC bright field is doing the deshifting. What I showed for the parallax deshifting, we did on quadrants. The TC bright field does a better job. It says every pixel is shifted according to its distance from the center axis. The shift of every pixel in the bright field region is proportional to the defocus, also proportional to its distance from the axis. This is the momentum transfer, the K vector. And they are correcting all of these K vectors. So it's in a sense like the difference between COM and DPC. We're doing a coarse correction. They're doing a finer correction.
Okay.
The shadow montage is something rather different. It's more like the tychography. I'm sure there must be a relation somewhere. I don't have a good answer to that.
Okay, then let's move to the next question by Professor Hong Zhao. He raised a question that the phage result you saw in movies are 4D STEM tomography reconstruction. Is that correct? And how do you deal with the beam divergence?
That is correct. And the beam divergence, the beam divergence is the signal in a sense. We worry about beam spreading, what we call beam spreading, which is the multiple scattering effect. This is giving us a kind of exit from the comfort zone at about 1 micron thickness. At this 200 nanometer thickness, even 600-700 nanometer thickness, we don't have a lot of beam spreading in the sample. Again, for the material scientists, you will find this surprising, but these are all very light elements. This is carbon, nitrogen, oxygen, practically. So we don't see, and we work, I should say also, we work with rather small convergence angles. So a few milliradian at most, in order to get depth of field. This is work in progress, but the beam, what we call beam divergence is actually the phase refraction on transmission through the specimen. So this gives us our signal in many ways.
Good. And then the next question raised by Stephen Oliver. The question is, how does the data collected by 4D STEM to be used in the subtomogram averaging in single particle case?
Yes, we have a long list of things to do.
Okay.
Yes, it's how can I say? It's on the way. But I would say that I would also say there are other labs now with the 4D STEM capabilities. Karsten Sachs for the DPC and Henning Stahlberg, also Angus Kirkland come to mind, who are more focused on the single particle approach. And since there are people there, I took the tack to tomography.
Okay, sounds good. Another question raised by Jose Maria Carazo in Spain. He said it's a great talk, and he has a question. that related the quantifications from a signal processing point of view. The experiment upscale by 4D or not? Something like local resolution of the images. In other words, do you have a spread of different resolution and different part of the images? I think that's these questions.
I can see an improvement in the power spectrum if I just rescale, but I don't have a good quantification. I'd say most of the quantifications that come in the cryo-EM field come after the averaging. When we speak about a 2 angstrom structure or 1.6 or 3.8 angstrom structure, it's in a single particle context, it's always after the averaging. So we've accumulated maybe 1 million images or 100,000 images in order to get to this combined resolution, and these are individual images. I think in individual images we don't see very much in a conventional method.
Yeah. Okay, a question raised by Professor Peter Lehman, and he thinks it's a fantastic talk. And the background filament you talk about probably phase DNA from his opinion, and you have also a lot of empty particles in the samples that may release the DNA. Do you agree?
I agree. I don't know what is in the background because I didn't notice the background until we did the upsampling. It was just kind of noisy stuff. There are empty phages. Let's go back to this image. They're mostly empty next to membranes, so they've kind of squirted the DNA out into the membranes, and we can see also the retraction of the injector, the stock. So these are empty. These are still filled here. Some of the filled ones have the retracted stock, so they haven't yet emptied the DNA.
Yeah,
this is all in
it's very interesting. You see a different name of the stock. You know, they probably are working at different stages of ejections of DNA. Yeah.
Yeah, we have here the injector in the middle, and the sheath of the stock retracts as the injector pushes out. I think Peter is really the expert in this, the world expert in this.
Right. Yeah, Peter is. Yeah, right. So Peter also have a question: would the CS corrector machine help for the 4D stem? You have the money.
We have a Themis microscope with a CS corrector, but in what we've done here, we're not even challenging the uncorrected microscope. So we will have to deal with that. The issue with going to higher convergence is the depth of field becomes very small. And so then we have to deal with different levels of defocus at different depths in the sample. And this is like the multi-slice tachography. This is another layer of complication and one by one.
Okay, then related to the last question raised by Professor Hong Zhao. He said one of the advantages of your techniques is to look at fixed samples. And would the beam convergence limit the resolution eventually? And in other words, I can't extend these questions. What do you think will be the theoretical resolution limit for half a micron sample? Have you done any calculation or estimates of that?
I don't have it at the top of my at the tip of my tongue. Here, I would estimate on the back of the envelope a maximum resolution of 8 angstroms because we have a very small convergence angle. We can go to a larger convergence angle, and then we can accommodate in half a micron. We can probably accommodate. I'm sure we can get to a nanometer. I don't think we can get to atomic resolution without doing this kind of multi-slice approach.
So if you do the multi-slice approach, you think you can get down to sub-nanometer resolution for half a micron material?
We'll have to see. That's where we go, but we have to see. It's a voyage. I would say just on the back of the envelope, the the lateral resolution is the wavelength divided by the numerical aperture, by the convergence angle. And then we can argue about a factor of 0.5 or 0.6. In principle, we can get to half of that. So we should be able to get down into the 2-3 angstrom regime. In principle, it shouldn't matter that we do TEM or STEM. I think we should be able to get to the same resolution, but we that we could get on the same microscope, let's say. But we have to take care that we are looking at unique structures. We're not judging resolution by the average after the fact, by combining the different specimens.
Yeah, so I think the real advantage, you can look at one sample at a time and can say something about it. That's very exciting. I think you really brightened the new year
Thank you.
method, and I think it should be more broadly disseminated to the cryo, the traditional or conservative cryo-EM community. And I thank you very much for this enlightening talk. Thank you.
Thank you.
Okay, I turn it back to Bob.
Okay, thank you, Bob. Thank you, Michael. It was fabulous. And actually, I had a quick question, if I may.
Yes.
Electron microscopy, one has to spend so much time at the microscope to set up all the right conditions for the imaging. And now with the 4D STEM and other approaches, there seems to be a lot of post facto analysis. I'm wondering what proportion of time do you think is spent between actually being at the microscope, taking the data, and then afterwards analyzing it afterwards?
There are two answers to that. One is the time taking successful data as a fraction of the time taking unsuccessful data. And the second is the time spent processing. So each data set can be 100 gigabytes. It's a heavy work to process. It can be a week to process an hour of collection. The question is which hour, and of course we don't know really which are the best data until we've done at least some of the processing. So it's heavy processing time. It's not a high throughput flow through just get the answer yet.
One approach is to have a global collaboration, so that will be working around the clock. I'm
open to that. I should say I'll be on sabbatical in New York for 6 months, almost 6 months, and I'll be in the States. I'm open to all kinds of discussions.
Okay, this is sunny California, blue sky every day.
We have blue sky except for.
I'm in contrast with New York. Yeah.
Okay.
All right, good. Thanks so much, Michael. Okay.
Thank you.
Thank you.
You can stop sharing your screen, Michael.
Yes, and I need to stop the.
Right, thanks. And before we go on, I'd like to thank Yi and Jane for setting this up again. So thank you, Yi and Jane, for this the arrangements today. And since our next speaker is from Trinity College Dublin, we'd like to introduce our future faculty member who will be joining us later this year, Kate Reedy, who got her undergraduate degree from Trinity and moved to MIT and Francis Ross's group, and in fact has spent some time also in Caltech. So she's probably knows people who are affected by the Los Angeles disaster right now. So I'll pass it on to Kate to introduce Lewys Jones, who's going to tell us about more about STEM than we would even imagine from Michael's talk. Thank you, Kate.
Yeah, thank you, Bob. And it's really great to be here. And it's really my pleasure to introduce Dr. Lewys Jones, who is the Usher Associate Professor in Ultramicroscopy at Trinity College Dublin, Ireland, which is also, as Bob said, where I got my undergraduate degree. So it's such a pleasure to see such fantastic microscopy coming out of Trinity. He's also the president of the Microscopy Society of Ireland, and his group focuses on methods for fast and low-dose STEM, event responsive STEM, as well as modular TEM hardware. So I actually thought it was a very good pairing today because Lewys really takes Michael's waste no electron motto very seriously in his work, as you'll see. And I had the pleasure of seeing him just last month at the MRS conference and have been following and admiring his work for a long time, both on the advanced STEM methods and also on the hardware side. So it's really exciting to have Lewys with us today. And thank you, Lewys, for joining us.
Thanks, Kate. Okay, so I press this and you should see my slides. Does that work? Yep, fantastic. Okay, well, thank you, Kate, for the introduction. Everything Kate said is true. Those are my many affiliations. I should just add one more by way of a declaration of interest. Some of the stuff I will show has now evolved out and is part of our university spin out. So I just want to be transparent about that. So this is what I'm going to cover. I will do my best to keep it to half an hour. Someone will shout at me if I go too much over. My approach to STEM is that all STEM is a time series, and I will hopefully convince you of that by the time I finish speaking. We'll look at picometer precision imaging in images and higher dimensional data. And then all of this stuff in blue is bundled under dose reduction strategies. So we'll look at single electron detection, the STEM temporal transfer function, and for people more familiar with a CCD MTF, this is the timed-based equivalent. We'll look at retrofittable digital electron counting, so how you can count electrons but with the cameras that you already own. Faster scanning, I think Michael already talked about the frame rate. I think people want for in situ, people want always more and more video rate. That neatly brings me to hysteresis measurement and correction, which has previously been the blockage to faster frame rate. Some tricks we do there using interlacing and scanning strategies to both improve frame rate, but also strain and spatial precision. And our current best efforts, which is around 40 frames per second video in the STEM. I'll introduce some recent work that we published earlier this year, which we call Tempo, which is measuring rate directly using even fewer electrons, and a multi-frame version of that. And if I have any time left, I will briefly show the research group's efforts at making our own 4D STEM detector head. I think everybody well knows where Ireland is. It's the now largest English-speaking country in Europe. And the AML represents a relatively large custom facility. And again, because Ireland is a very neutral country, we have Zeiss, Neon, Thermo, Joel, every possible vendor of instruments. So we get no group discount negotiating our service contracts. The email for the core facility staff is here, and people are welcome to email if they want to visit any of these instruments. More widely, my group kind of has three areas at the moment. One is physically making pole pieces and hardware electron optics. Increasingly, this is for the life sciences, for single particle cryo and high throughput connectomics. Electron detection, which is mostly what I'm going to talk about in fast STEM. And the last couple of years, we've been very interested in moving towards a net zero energy TEM, which sounds a bit fantastical, but we think it would be possible within the next few years. Okay, so Michael has saved me a wealth of time here. There's a microscope. There's the same view of the STEM, greatly simplified. We make a focus probe, we scan it around. Because the beam is scanned and the data is collected pixel by pixel, everything we do is formed with respect to time. It is a convenience for the way that our eyes work as humans that we choose to arrange that with respect to space in a grid of pixels. Any deficiency between that assumption and reality is where STEM gets a little ugly. And so that can take many forms. So this is a work from now nearly a 10-year-old work of mine, where I'm plotting here the different spatial frequencies of various flavors of STEM distortions. Simple translational drift, that's just called drift correction. Translation is the easiest one. The next frequency up, we get into sort of a fine transformation where the image has a skew from a slow drift within a frame. The very fast stuff, it was scan noise, which, I spent my early part of a PhD on this. Line sync at 50 hertz, or in the US 60 hertz. This is mostly dealt with in power systems. But this stuff in the middle is the really insidious stuff. So a fine transformation is fairly easily dealt with. People in the AFM community were on this years before us. Scanning from top to bottom with a stage drifting to the right, you get an image that looks like this. Scanning from bottom to top, you get an image that looks like this. And they're mathematically equal and opposite. You can take a pair of frames and back out any linear stage drift. This stuff is more problematic. We have a very relatively slow scan line in STEM. So between the lines, there's an appreciable time. Along the line is relatively fast. So this panel here is the most common kind of distortion we see in the STEM, where there's a variation between the X and Y vector from row to row. There's been work for correcting this, colinophus with perpendicular pairs and so on. And the group of Jim Lebeau scanning around the clock face in maybe 10-degree increments. It works well. Each of those is in a fine transform, but it's slightly challenging to publish circular images. But all of these things can be dealt with in designing the collection and then post-processing. These are some lovely examples of approaching picometer precision from multi-frame imaging. So works here from Spain, Norway, this is Belgium, where we have around a picometer standard deviation either side of this very subtle domain wall. These are several examples in imaging where the precision was explicitly evaluated, but the same is true in spectrum imaging. So this is sort of 3D data, multi-frame tychography, which is either 4D or 5D, depending on how you do your counting, where because of the slowness of the cameras, all of these periodicities are moved down about 2 or 3 orders of magnitude. So what used to be an affine transformation, simple to correct in imaging, becomes hard to correct in tychography. But this is monolayer graphene. And even in another dimension, a precession dimension, segmenting SPED data and then reconstructing to improve spatial precision. So this has dominated a lot of my research for the last decade, multi-frame STEM of different flavors. The problem with multi-frame is it puts multiple dose into the specimen. So then the biggest question is, okay, we need to reduce the dose, and then how low can we go? So it's good to revisit this equation for dose. If Ray was here, he would tell me I really should actually say fluence, which is more accurate. Dose is a different unit for biologists, but we're a bit lazy. To do this, you just need to know the Prohm current, the Coulomb charge, the dwell time that your probe is on each pixel, the area of that pixel. Here I'm showing a square pixel, but it doesn't have to be square. And then if you're doing some sort of interlacing, compressed sensitive or adaptive sampling, some sort of fraction of the pixels hit might be in there. Why am I showing a child on a bed? Well, in general, one should want to use the minimum radiation to get the job done. The operator tends to be well protected. Our specimens tend to not be well protected. So minimizing radiation is always a good starting point. We can increase the pixel width to a point. If we drop below twice Nyquist, we just won't have sampled the features we want to look at. Everybody can hopefully reduce the probe current, either with monochromators or extraction changes. We can reduce the dwell time up to a certain point, and then things get a bit squirty. We'll look at that with some examples presently. And later I'll show some examples of adaptive sampling. My group is not doing any work in compressed sensing, but we think adaptive sampling has some good benefits. So when you want to use every electron, as was said in the introduction, it's important to observe every electron. These on the right are 3 literature results from other groups, Rosenhauer, Lebeau, and Yannick Meyer, going to very low beam currents, where on the Hadouf detector, individual electrons started to be visible, appearing one by one. Unfortunately, it was too difficult at that time to form an image. The closest was this Mittelberger paper, but using the electronics that come by default with the instruments, it just wasn't possible. In the recent years, this PhD student, now Dr. Malarkey, collected several detectors that we could get our hands on through our own lab or collaborators, and plotted their rise time and fall times on this useful chart. It's very helpful. So why do these single events have rise and fall times? Well, the detector is a scintillator. And what that means, which when you go to these very low doses that people often don't expect, is that there is this really long afterglow. If I tell you that a phosphor on your watch face has a long afterglow, you're not surprised. If I tell you the scintillator on the detector bleeds intensity across the entire width of your image, you would go running to your service contract and want to complain. So this is a sort of fib lamella sticking out into vacuum, and there's all this light bleed from the scintillator onto the STEM detector through the vacuum. There's a fast response of the order of maybe a microsecond, 1.5 microseconds. There's also a slow response of several microseconds, maybe 10 microseconds afterglow. The afterglow is so long, it actually wraps back round to the start of the frame, and you can see the right-handed glow of the pillar even on its left. What this looks like in a high-resolution image, as you go faster and faster in your dwell time, you see streaking in real space, or what looks like a band pass in Fourier space. If that was two-dimensional, CCD TEM guys would expect to see that as an MTF, because it's one-dimensional and only limited by time and not pixelation. Instead, we call it the TTF, the temporal transfer function. I'll skip most of this, I think, because my postdoc can explain it better, and it's published here in ultramicroscopy. But we see, for example, for slower decays, we have a more aggressive damping in the transfer function. What it looks like maybe more intuitively are some practical examples. Here we see experiment and simulation. Our simulations in the group now incorporate these scintillator afterglow effect to model the detection properly. And as we go faster and faster and faster, this MTF is shrinking in. So what this means is we can use this TTF as a planning tool to know for any given detector what is the fastest frame rate you can safely go at before you start unacceptably compromising spatial resolution because of temporal afterglow. Okay, so that's where we were until recently. Of course, it's not just enough to document what the microscope is doing poorly. One should hope to try and fix it. So to do this, we hooked up high-speed oscilloscopes to our detection elements, and we're looking here using a high-speed oscilloscope. We're looking inside STEM pixels in the time domain. Here, as I said, we can see these individual events arriving. Some of them are stronger peaks, some of them are weaker, some of them arrive very close to each other, but there's nowhere on this voltage axis where we can put a threshold and see all these events. For example, if I put a threshold low enough to detect this event here, both of these events will appear as a single event. If I put the threshold high enough to not see these as single events, I miss this one completely. However, in the gradient domain, what these events all have in common, although their heights are different, is they all have sharp rising edges. So in the gradient domain, it is much easier to choose a threshold where I will see every single event and then digitize this signal. Even these 4 events, which arrive very quickly one after the other, become cleanly separable in the gradient domain. Now, it's too difficult to do this in code. You have to do this in hardware, and for that we use high-speed FPGAs close to the microscope detectors. That's represented in time. In the image space, it may be a little easier to show. So this is just a very low-dose STEM image recorded incredibly fast, where we're now seeing the temporal afterglow of every electron. We put that through our digitization hardware, and this is the image we get. So this is around what was common in CCDs 10 to 15 years ago, going from analog CCDs to electron counting cameras, but we've not changed the scintillator head. This is just the color mix. We're using the original detector that you already bought and paid for, and just changing the electronics that sit outside the vacuum. So this is how it's connected. We have the signal coming off the detector raw. We just put a T-piece in that. The original analog signal can go to your scan controller. Could be a digiscan, could be an ultrascan, could be the T-VIPs or point electronic. And another T goes through our pulse counting system, which then sends a digital input to, say, the digiscan. You get a second image. The microscope thinks it has 2 cameras now. You get 1 analog and 1 digital. And you can go down to hundreds of femtoamps of beam current and still retrieve usable images. This is my disclosure. This is the unit that is now available through the spin out from the university. Going forward, we want to do this on multiple segments. And this is a photograph of the Elbaum opal detector, where we want to do this in DPC. Since we have biologists on the call, I thought this is nice to show a biological tissue. I'm not sure what it is. I think it's a human macrophage cell, but I could be wrong. The analog image looks quite nice. It looks sort of softer, looks smooth, but actually that's because it's been smeared in time. You see the damping envelope in the Fourier transform, and there is just resolution has been lost from some of these fine features that you cannot recover. The other nice advantage of this is that we naturally get digital data. The signal is quantized in integers, and that is very useful because that's what our computational colleagues, image simulation people are doing. It's what the machine learning guys are doing. they are training their models on digital simulations. Now, for the first time ever, we can actually give them digital counted STEM experiments. And there's just 2 example works here, one from the San Vito group in Trinity and one from Ishikawa in University of Tokyo. And in both cases, it seemed like the digital images were easier to denoise because you can give the computer some prior knowledge about Poisson distributions and things like that. So in my time series earlier, I showed you that some electrons had very large peaks, some had very small. This effect arises from what we call detector inhomogeneity. And this is a work that we published 10 years ago now called How Flat is Your Detector? Again, we've anonymized these, but you can probably pick out your own one if you've ever looked at this, where there's big variations in the efficiency. In this experiment here, we just scan our beam across the detector and record the image of completely homogeneous beam current being recorded in the intensity of the frames. This is a very famous, very ubiquitous detector that has a big dark shadow here. So an electron striking the active surface here is recorded about 3 times more weakly than one striking out here. When you go digital, though, this flattens out. An electron is a 1 or a 0, so just summing together 100 frames here at low dose, we can show that this inhomogeneity is largely resolved. It's not perfect because there are still some losses. And that, hypothetically would represent some of these weak peaks and some of these strong peaks that were recorded. So now we've shown that we can lower the beam current and scan fast, observe single events and count them as digital to get sort of a perfect DQE in a way. That was good. We enjoyed that for about a year until we realized that scanning fast has its own problems. The scan is not just a simple raster. Okay, we have interframe time, which is lost. If you're using flyback weighting, you have interline time, which is lost. If you're doing line syncing, there's another lost time. If you're synchronizing to an external camera clock for, say, 4D STEM, there's another lost time. And all of these losses add up. It's all exposure that your specimen receives that didn't produce useful data. So just as an example with a few different flyback times here, somewhere like 600 will be very common in many labs. At the pixel times that we want to go to, deep results below 1 microsecond, maybe the beam is only useful for 1/3 of its illumination time. That's an efficiency problem. We also have a fidelity problem because the scan doesn't, well, if you ask for a scan that looks like this, what you get is something like this. So this is the work of Sang, and I think this was Andy Lupini on this one. About half of the frame here is unusable because of this distortion. We usually hide that with flyback waiting time, but it was sort of there, but not shown to us. So where does this come from and what can we do about it? Well, by and large, the scanning technology we use hasn't really changed since one of these. For the younger people on the call, this is a television, which at the back of it has coils not dissimilar from what's inside our stem. Perhaps the state of the art is this twin wound coil. So this is a coil published about 5 years ago now, a collaboration between the University of Tokyo and Yale, where they had 2 different scan coils. There are 2 scan coils in here because one is strong and slow, one is weak and fast. So this coil is used for low magnification imaging at low frame rates, and the smaller coil is used for high magnification imaging at higher frame rates. Why is the coil slow? Why do I say the coil is slow? Well, it comes from the fact that ideally in a wire, you would like to ask for a new current and it would immediately change. Current in our wire is proportional to our deflection of the beam. In reality, we have this decay from the infinite inductance of those coils of wire. This is a cartoon, but we have actually measured this. So we've hooked up oscilloscopes to coils in our lab. This is what it kind of looked like. And you have here something 30, 40 microseconds at the end of a line until the beam is ready at the start of a new line. That's quite a long time to be waiting for your beam. That could be hundreds of pixels available to you in that time. In an image, what it looks like generally is this sort of compression. So here I'm showing just some cropped letterboxes from images of STO. STO, not much unknown properties about it, but we use it here as a sort of picoscale ruler. We know a single crystal should have perfectly equal unit cell spacings across its width. And here, going faster and faster and faster, basically eliminating our flyback waiting time by the end, you can see this compression on the left side of the image. you know, to the extreme case that we're looking at sort of 15, 17% strain. There's no strain in our single crystal of STO. It's purely an imaging artifact. Fortunately, it has a very reproducible exponential decay-like fit, and we can correct for this computationally. So we can unstretch the images before we align and average them. We discovered this when we were trying to replicate this sort of Lewis work in our lab here with very short flybacks. The Lewis work works very well for long flybacks, conventional flybacks. When you push it down, all four image edges become corrupted. You can't cleanly average this or publish it. I mean, you could just crop out the middle, but then you've wasted field of view, dose, and frame rate in doing that cropping. The solution was to individually correct the frames scanned in these different directions and then do the alignment and registration. So that works. We publish this and the codes and how you can replicate this on your own instruments or in the supplementary information. But that still doesn't, you know, that doesn't really feel very satisfying. We would like to fix the instrument at the hardware level. And so I said, you know, the work from the University of Tokyo has the option to use either low magnifications at low speed and high magnifications at high speed. But what if you want, for in situ, say, or biology, low magnifications and high speed? So why do I have a leopard and a horse here? Well, so the leopard, or the cheetah, will accelerate much faster than the horse, but quickly run out of stamina. This is like the small coils. The horse will take longer to accelerate, but it has a lot more long-term strength and speed. So what we have started developing here with a PhD student, Adam Phipps, he's running 2 separate scan coils with 2 scan controllers and 2 power supplies. Basically, all of your fabrication electronics costs just double. But this is the rig. These are the first coils he made. We've got 3D printed mandrels, a little counter so that the student no longer has to wind them by hand, our own power supplies, and then a jig that roughly replicates a sort of TEM-like geometry. And there's the slow coil and the fast coil. And what this looks like then, and these are real experimental measurements, we have the slow decay from the main strong coil, fast response of the weaker coil, and by combining them with bespoke software, we can push that previously 40 microseconds until the line is usable down to about 14. So we can get a more than half reduction in the flyback waiting time. And that leads to better efficiencies, better frame rates, which I'll show in a minute. We can also do some other tricks from the days of early television, which was interlacing. This was very common at one point when you wanted to get more frame rate for sports broadcasting, but you didn't have the physical bandwidth. We are bandwidth limited by dwell time, and we still want more frame rate. So you can record these sequentially, the odd and even lines, and rebuild the data later. Interpolating the missing lines is relatively trivial. And we've shown, this is simulation on the left and experiment on the right. Again, we're using strain maps of single crystals to infer precision. And going from conventional full frames, that would be sort of Shannon scanning, to rotation, interlace, and then interlace with rotation, we can get about a 2x improvement in strain precision, or if you prefer, about a 4x improvement in dose efficiency at the same precision using these sort of interlaced approaches. And it seems to work in real experiments as well, not just simulation. This is made much easier if you have a programmable scan controller. And of course, once you have a programmable scan controller, you start doing all sorts of other mad things. Here's some examples showing Hilbert scanning, serpentine scanning, spiral scanning, and pretty much all of these work equally well as Shannon scanning when you're at this level of, say, 10 microseconds per pixel, fairly conventional. But the frame rate is poor. The problem happens then is, well, okay, so let's turn our scan generator up to 11. This example shows interlacing using the method on the previous slide. We got here a 300 by 300 pixel image. This is using a 18-year-old Titan G2, and we can hit here about 16, sometimes 17 frames per second. This was just melting some sputtered platinum palladium on a dense heating rod just to give us something to look at as a movie. But 17 frames per second on a nearly 20-year-old stem, we were pretty happy with at a field of view which is I think useful to in situ people. Okay, so then we say, well, let's go faster, and we hit that finite inductance problem. So here the Hilbert scan is kind of crystalline, but largely gibberish. The spiral scan looks good in each individual quadrant, but then there's a big distortion when the beam changes direction along these diagonals. And then the serpentine scan looks like a double exposure of this waffle, where the two parts of the image have been shifted because of this inductance when we turn around. So these first two are quite difficult to deal with. This third one, though, is a bit easier. It's reproducible. The beam is always traveling in the same direction across the whole stem line. So what we can do is we can do what we call an offset or overdriven serpentine, where we pre-program the scan path we ask for in our programmable scan generator to overdrive and compensate for the lag, so that then after the inductive lag, we're back with the probe where we wanted it to be, and the image then comes out normal. And so this should show that. So this is a 512 by 512 image running at something like 40-ish frames a second. I don't have the exact number. 47, I think, was our record. This is unpublished, but you can see here, the postdoc is screaming around at huge magnification across the entire stage of this waffle, and at no point do you lose tracking of the sample with the image being torn. Now, it's true at this pixel time, the pixel time here is 50 nanoseconds. With a pixel time of 50 nanoseconds, we are seeing that streaking, but then we can fix that next time with the digital readout of our individual electron events. So using that, we can hit 45 frames per second with digital detection. Okay, so there's all the time bringing the dose down, making the microscope easier to use at high STEM video frame rates. What if we want to go lower than having the beam on every pixel? So probably most of you in your offices, homes, in your cars, you may have LED lights. Many of them may be dimmable. LED lights don't ever actually dim. They just turn on and off for a different portion of their time, what's called the duty cycle. So a 50% duty cycle will just be the LED on half of the time and off half of the time. This same technology was developed and then implemented by IDES for the jail platform. There is a high-speed shutter up here just after the gun, where this now switches on or off in about 10 nanoseconds. It's a little bit faster, but switching either on to off or off to on takes about 10 nanoseconds. That means that this can switch on and off many, many times within a single dwell time, and can be used for this pulse width modulation dimming in the stem in real time. And why is this important? Well, so as Kate was talking, it's often important to measure the weather in Ireland. And I want us to think for a minute about how we measure the rainfall. So the normal way that you probably learned in school is you take some sort of cylindrical beaker, you measure for a fixed time, say 1 hour, you measure the quantity in the beaker, millimeters, and then you plot a graph of millimeters per hour for your primary school science homework. If you go and measure the rain like this, you will get soaking. If we expose our samples like this, they may get destroyed. A much better alternative is what your child would probably do if you ask them, is it raining? They will go outside, start an observation, maybe put their hand out the window or something. Look at the time it takes to get 1 drop. Maybe collect 2 or 3, the time it takes to get a few drops. And in their mind, you will invert that to an event rate after only a handful of events. Once you've got the event rate, you just go inside or close the window. You no longer need to keep measuring again and again and again the same event rate. Now, by combining electron pulse counting and fast blanking technology, we can achieve this for electrons in the stem. So this is what it looks like in practice, and this is experimental data from an oscilloscope. These are our analog pulses. It's not quite as fast an oscilloscope as the one that we use on the FPGAs, but this is the analog signal, then the digital signal. In the white and gray stripes, I'm showing the pixel times, and the last row is the green being down means that the beam is open, green being high means the beam is blanked. So the pixel starts here, we open the beam, events start arriving down the column, and after 2 events, we send a signal to blank the beam and shut it for the rest of the pixel time. No more events are detected. At the start of the next pixel clock, we open the beam again, 2 more events arrive, we close it. Again, 2 more events arrive and we close it. In all these pixels, 2 electrons reach the HADF detector. If it's a long time, it's a slow rate. If it's a fast time, it's a high rate. We're forming an image now with 1 or 2 pixels being on the sample, being on the detector in each stem dwell time. This is like normal STEM, a Poisson process, and if the collection number is higher, the uncertainty is lower. That's unsurprising, and it behaves just like any other Poisson process, but with a great dose saving at the specimen. What this looks like in the image, so this is a plot of the time to trigger, here waiting for 2 electrons to arrive at the ADF. And it kind of looks a bit like a bright field image, because on the atomic column, the time for 2 electrons to be scattered from the probe to the ADF detector is low. In between the columns, that time is high. So this is just simply inverted, and we have directly a scattering rate map in our ADF-like contrast. This is an example of silicon nitride, just proving that you get a normal ADF-like contrast. Average a bunch of frames here if you like to just increase the signal to noise. You can also do it with bright field, but you might select a higher threshold than with the dark field. We've done a comparison of the scattering rate with conventional pulse counted STEM or event responsive. In the high dose limit, they do trend to be 1-to-1, so the type of contrast you're getting is exactly the same. The noise profile is very slightly different for aficionados, because in the event responsive case now, every pixel has received the same dose at the detector, not at the specimen. So we tend to see the bright columns a touch noisier than conventional, but the weak columns less noisy. And this is very good for light element imaging. Biological tissue is what we normally go to when we want to test how fragile something is. And the same is true in STEM. So here I'm showing a tableau of images with increasing trigger numbers. That's the number of electrons where we blank the beam. And actually, we can form an image with contrast where every pixel in the image received 1 electron. And the contrast is now coming purely from that timing signal. As we increase the dose, the signal to noise improves, and that's not surprising. And we can plot that out here using a sort of Fourier ring type approach that's more common in biology than physical science. Our duty cycle and our dose scales linearly with the number of electrons we wait to count for. And what we can do, we can also then take the signal to noise divided by the dose and get a plot of the detection efficiency, the extra resolution we're getting for the given dose. And what we find quite routinely, this tends to peak around 4 to 5 electrons in each pixel. And that's sort of somewhere around this level of contrast. Now, a human will obviously say this looks better, but to a computer scientist, and maybe you're looking at doing it in a burst movie of these types of frames, a movie of these frames is more efficient than five frames of this would be more efficient than 1 frame like this. There's another example for the physical scientists. This is a phagocyte type zeolite. We have an average dose of 370 electrons per square angstrom, and we can see at atomic resolution these Si aluminium columns here. Now, at the time, we had the dark field detector in, and I really wish we'd had the bright field, because this is a zeolite image at atomic resolution using dark field. This would have been so much better if we'd have used ABF or DPC, but we just were trying to get data so quickly we didn't even insert the detector. So I mentioned very early on, what about other alternative scanning strategies? And what I said was adaptive sampling. Well, we can even go further than this event responsive mode now, what we call tempo stem rescan. This is an example of a tempo image with a trigger number of n equals 3, and this is just a zoom in of this little patch. Now, we know our stem has a finite point spread function. We don't really believe that salt and pepper noise, the big hot yellow pixel here, is probably not real from our point spread function. Well, that's actually a kind of prior knowledge that we can exploit in real time on the instrument. So we could do something like a Gaussian filter, subtract that from itself, and we see the high and low local deviations, or salt and pepper, sometimes called noise. We can look at that and say, well, let's take the more than 1 sigma outliers, and these are the pixels where we probably would like to narrow down that Poisson uncertainty distribution. And then we only rescan those pixels. That means that the probe is visiting maybe 1/10 or less of the locations on the next pass, because we were already happy with the measurements of the others. What does this look like? So we've done this now experimentally on a Joel F200. This is the enlargement from that data there. And when I arrange the data in frame number, this rescan appears to perform very poorly. However, in this rescanning, the fraction of pixels revisited falls very quickly and continues to fall as the noisy pixels update themselves. This is about 90% after the 10th frame. So when I use this and put these on an isofluxual sort of axis of the cumulative fluence, it looks like this. Very, very roughly, if we set this limit of some resolution criteria in arbitrary units, we can get there with less than half the dose. And this is by, in real time, in between the times of the frames of one of these fast movies, determining and exploiting that rescan mask and only revisiting what we need. It works with spectroscopy, but it's a bit more subtle, so I don't really want to go into that. But for example, say in EDX, we have the same sort of signal to noise for light elements as heavy elements because it's based on the photonic detection rather than putting the same beam in every pixel. Okay,
should
I'm
probably
probably
be
just going to use
wrapping up soon, Lewis.
I am going to finish, Bob, I promise. Just because 4D cameras were mentioned, and I'll mention at every meeting I go to. This is a plot from a review article I did with colleagues showing the explosion in data from 4D technology. Every time I hire a data scientist, I don't hire a microscopy with that salary. I would like to hire more microscopists. But there's previous works from the Oxford group showing that around 8x8 array elements is fine for tychography. Our own work shows the same with DPC. And this falls in a really interesting space. So from this other sort of review tableau we published last year, the Arena, as Michael mentioned, is the fastest 4D camera that I'm aware of you can buy commercially. This is the danger zone of many pixels on the camera and very fast pixels. Nobody is there, fortunately, but people want to keep going there, it seems. And then the STEM detectors are down here, very, very fast, but with one element. And they're gradually moving to the right. And they have these two communities, cameras getting faster, STEM people adding more elements. And there's this whole thing in the middle that's currently underexplored. So we've been addressing this here in Dublin. The first stage was actually flange design. We're encouraging all our colleagues, especially in Europe and in Germany, there's a lot of interest in standardization. We've created all of these adapter flanges so cameras can be developed on CF flanges and moved between all the different microscopes in the building. The first one of these that we've made and tested is here on our Joel 2100. I'm happy to say this is holding vacuum now, so our technical staff are happy. We've been using low-cost silicon photomultipliers. Here they are on an evaluation board in an old Zeiss Evo SAM, and using relatively low-cost technology, we can detect single pulses in a way that people can build a camera DIY in the lab. So we're aiming for that sweet spot of speed, time, pixel number, and single electron counting sensitivity, all with stuff that you can use and build yourself. So I just replaced outline here with conclusions. So I hope I've convinced you that all STEM is a time series, including the deficiencies. I hope I've shown that you can get picometer scale precision in imaging and spectroscopy. I've shown single electron detection with the detectors you already own in your vacuum right now. The implications for that on the temporal transfer function, and how we solve both of these problems with indirect direct detection, replacing the electronics. We've gone faster. When we went faster, we had hysteresis. We overcome some of that with hysteresis, with interlacing and novel scanning strategies. We can get over 40 frames per second with a 512 by 512 for in situ colleagues. And then these tempo stem and tempo rescan to really do event responsive imaging using every electron most sensitively. And come back in 3 years and I might have something to show from a working 4D camera made in the kitchen. Thank you. I should leave these acknowledgements on the screen because many people, this is my research group, it's a year old now, but the guys at Joel and Cambridge and locally. Thank you.
Thank you, Lewis. I think, Kate, you are the host.
Oh, of course, yes. So we'll take some questions from the audience now, if there are any. People can feel free to raise their hands. In the meantime, I also had a question, Lewis. So when I see your work, I think of it kind of in the context of in situ, or how we could use this to do some cool in situ studies. I wonder, has the event responsive aspect ever been thought of in the sense of, sometimes when you're starting an in situ study, you're looking at like very low mag to begin with, and then you want to see something happen and immediately zoom in on that area a bit more. Would there ever be anything that you could envision where, maybe if electrons are counted in a particular area, then that can be noted as the area to like keep scanning for a longer period of time? Does that question make sense?
I think so. So one of the things we do, Kate, is there's a couple of things we do here in the group. One of them is that with scripting, we buffer everything that the user sees during searching. And that's stored in like a sort of, I think in America you call like a TiVo kind of box. So you can always replay and watch that back. The second thing is that, you know, in the past, operators have been used to this kind of like search, preview, record kind of language where we put more time and dose as we move through. Well, actually with tempo, you don't really have to make that choice in dwell time. The dwell time can remain constant, and you can move up in the trigger number. So if I show, let me see if I can get back to this in a more sensible way. This one. You can search and navigate with a trigger number that's very low, and then switch to a higher trigger number if you see something you want to record. So you can minimize the exposure when you don't want it, maximize the signal to noise when you do, and all the time your dwell time and frame rate can remain constant. You know, so if you're timing a melting or whatever, those processes remain constant, even though the dose can respond dynamically.
I see. So, but would you have to so say in your in situ video where you had the melting of that thin film, could you program it to say, you know, once you see a movement, begin to increase the N number?
You could. I haven't. I mean, I'm reminded of things like what is it? The prototype software that sort of sends and receives commands based on things happening. I mean, I think, you know, with a sort of an API into the equipment, yeah, you could imagine that. Yeah.
I think. No, it's really fantastic for in situ. We would be excited to try and use them.
Well, there's also another alternative which I didn't show, which is what we call skip ahead tempo. It's more difficult because you need to integrate it with also your scan generator and a clock input. But instead of blanking the beam, you do have the option of just skipping to the next pixel with the probe. And there, instead of a 90% dose saving, you'd get a 10x frame rate boost. It's a little tricky because you're getting the same sort of probe hysteresis issues that compressed sensing suffers from, but it's not impossible, just a bit challenging.
Okay, I see.
So we should be wrapping up fairly soon. I don't see any
We also had a question from Michael. Michael, I don't know if you want to unmute yourself.
We definitely want to ask Michael for comments, and there's a question in the Q&A from Thomas Dasher. If you got that, Kate.
Yeah. I'll wait.
Okay. Yeah, Michael sent his question directly to me. So we had a question from Thomas Thursleff, which was, for the temp rescan, how does the sample drift affect the fidelity of the spatial registration for later frames?
Yeah, that's a very fair question. So at the moment, we've only demonstrated it at relatively low magnifications. This example here is I think is a evaporated test specimen. It works really well on things like our neon ultrastem. Where there is other drift, you want to look at drift tracking and drift prediction, and that's where the research is going now, to increase the available magnification for the rescan. But yeah, at the moment it's working well. Low mag biological tissues or stable microscopes, and there's ongoing work for the other areas.
Great. And now, Michael, I'll let you unmute and ask your question.
I was wondering, what is the dynamic range when you have this event detection? So if you can certainly identify the individual ones, but they're smeared together if you go too fast. So how high a current?
Yeah. So that's a very fundamental question. If we go back, right back. This one, yeah. This is a exact analog to what EDX spectroscopy people would call event pileup, and you get dead time on your EDX detector. The great news is it forces us to low dose. So you want the events to be arriving something like around 1 per microsecond would be fine. I mean, this is not really comfortable. this is 5 microseconds here. 1 per microsecond would be great. That's the sort of rate we're looking at there. More than that, and you start to really have risks of coincidence loss. You can improve that a bit with faster scintillators, direct detection, tuning your preamps, or a detector with more segments, because your coincidence loss is only on a per segment basis. So even, for example, like the Elbaum opal with 4 segments on the brightest ring, you get 4 times the dynamic range. So we're very interested in segmented detection from a dynamic range point of view. The good thing is, once you go to that point of coincidence pileup, you can go back to analog detection and you're still in a Poisson regime. So it's nice, and that's one of the reasons why we tee off these detectors, because by teeing off you have the digital and the analog, and when the digital and you can actually see it. We've graphs of this that should be published this year. The coincidence, the beam current, it goes up and then it falls down when you have the detector saturated. So it's very easy to diagnose and avoid. But yeah, forcing users to use lower doses, I don't really consider a negative. The students should take it up.
That's where the life science comes in, because if you don't use the low dose, you see a big hole.
It strikes me that there's going to be different scanning strategies for different applications, from the biological applications to in situ imaging, to the best picometer or sub-picometer if one wants resolution of the images and the atomic positions. So it's going to take a while for us to figure out what's going to filter to the bottom or the top as terms of strategies. But I think that having multiple strategies like this available to us really expands the use of STEM in the future. Michael, what's any comment from the soft from the soft materials point of view, or biological materials?
I think we've come to many of the same conclusions, but not we haven't driven it as far. We did we did a project that it's in kind of on the line to be resurrected, but it was scanning in circles, and my preference for scanning in circles is that you don't kick the ground plane every time you hit the end, and you give it a triangle wave. You hit the end, and hit the end, and hit the end. There's a pulse going through the whole electronic chain somewhere. Then you have it's like the middle is twisted, the nose is twisted right in the in the middle of your face. But Chacha actually figured out how to untwist this on a fundamental level with solving the induction equations. So then it comes out straight, and this was a thought for going to the single particle approaches. But it's
so, I mean, so Michael, we discovered things like the inductance issue imaging, high resolution single crystals. We're implementing this now, these multi-coil geometries. The hope is then they will trickle down to all kinds of instruments, and even when you can't always see that little compression, like say on a tissue, it will just benefit everyone, because we hope that the coils will adapt, and that nose you described would just disappear, we would hope.
For sure. It's kind of shocking that you work out this timing and you see that the beam may be 100, 200 microseconds behind the place you tell it to be. And you see this when you scan slow and fast, which we do for the low dose procedure, slow and fast on the same area, and there's a big shift to the left in the raster scan. So if anybody tries this from the bio side, don't be alarmed. That's normal, unfortunately.
Well, that's what we saw, that double exposure of the waffle, but in the interlacing,
Okay, I think this is really exciting development. You have all the tools in your toolbox, and I think our biologists have a lot of problems of biological natures, and I think we have to keep talking, not just talking, but doing as well. So I think it just launched a really exciting year that we can work together in some of these problems and solve. Hopefully we have a whole recipe, depends on what is in the kitchen. So, all right, I thank everybody joining us today, and wish you the best in the coming year.
Thank you. Thanks, Kate, for your hosting, and Michael, Lewis, for some very stimulating thoughts. Yi for your help, and Jane. and Wa for great collaborations and a lot to do in the future. Looking forward to these developments. So thank you everyone for joining us, and see you in 2 months' time. The first Monday of March would be our next EMX.